Sex-linked differences in responses of gut microflora to diet by Group 5, Molly Butler, Carol Tong, and Lillian Chen.
The human gut contains an array of microbiota, a large diverse population that aid in digestion in the host. This symbiosis is mutually beneficial as the microbes digest food that humans cannot digest on their own, and the microbes gain nutrition and shelter from their host. Things such as genetics, environment, and diet affect this population in humans, and in turn this population can have varied effects on its host, including effects on the health, nutrition, immune response, well-being, and metabolism of the host. In some cases, however, it is not clear what is influencing what, like in cases of obesity and diabetes, in which there is a correlation to low biodiversity of the bacteria.
Scientists then took measurement of isotopic carbon and nitrogen ratio from the fishtail mussel. Carbon isotopes are fixed in different ratios by different primary producers in lakes. Because the fish diet contained two types of primary consumers feeding on different primary producers, the carbon ratio found in fish, comparing to the carbon ratio in mollusk snails, could be used as an indicator for the type of diet they lived on. They also tested the bacteria DNA directly from the intestine. 16S rRNA gene was specifically used for identifying microbes. The genes for individual sample were amplified using polymerase chain reaction, and the result DNA sequence was analyzed to build microbial taxonomy and was compared to ones found in other individuals and other sexes. Variation in microbial species abundance between sexes was calculated and used as a measurement to confirm the existence of sex-diet interaction.
Researchers found that in evolutionary divergent species such as the three-spined stickleback and the perch, the trophic level at which they eat does change the overall composition of the gut flora. There are also differences between the diet and sex effects on the microflora within the host body. This means that diet as well as the gender of the host work together to alter the effects on the composition of the microbiota flora in an individual. There is significant difference in such that diet and sex both influence the type and number of each microorganism present in each species. Although diet effects on the gut flora between species, the stickleback and perch, are positively correlated, the same does not hold true for the comparison of gut flora between the two sexes in each species, because sex does influence OUT abundance in each host. The reason why there exists differences in gut flora between hosts of different sexes in the same species are not entirely known, but scientists are currently looking at hypotheses that might explain this phenomenon, such as interactions between hormones or the immune system with the gut microflora. Scientists extended the research further to include two other mammalian vertebrates, humans and mice, to test if the findings of the sex diet influence on gut flora applies to them. The results yielded that sex and diet did in fact have an influence on the type and abundance of particular strains of microorganisms in the hosts. In mice, for instance, lactobacillus increased in number in male mice that were fed a high-fat diet instead of a chow diet. However, the opposite occurred in females, where the number of lactobacillus actually decreased after changing to the high-fat diet. In addition, for humans, once researchers reanalyzed previous studies done on the gut flora again, they noticed a significant abundance of sex-diet interactions, particularly for Fusobacteriaceae. In conclusion, the diet and sex interaction on the composition of the host gut flora holds true for all four divergent vertebrate species. The three main points to remember from this article are: 1. Wild-type species have significant variations in their gut microbial composition between each individual in the population. 2. Diets of individuals affect the microbial differences among individuals within a population. And lastly, 3. Sex plays a role in the effects of diet on gut flora.